Everyone. In the previous part, we had gone through mushroom importance and types. Now, in this part, we will discuss cultivation practices mainly employed in variety of mushrooms like your Agaricus bisporus, Pleurotus australis, Volvarelle, Volvace. Okay. Now, let's discuss about the first type of mushroom cultivation, that is related to the Agaricus bisporus. And you all should recollect this Agaricus bisporus is commonly called as button mushrooms. Okay, next. So just recollect it. The Agaricus bisporus is an edible basidiomycetes mushroom were cultivated in European and North America. Nowadays it is being cultivated in India also. And the other common names of Agaricus bisporus, or these all common, cultivated, table, champion, all these things. But in general, it is being referred as button mushrooms. Now, these are the cultivation practices or the steps that are being followed in the cultivation of Agaricus bisporus. So here, see, the first step is production of mushroom spawn. Then what is meant by this spawn, I will tell you in detail. Then substrate, that means what are the bases that are required for this cultivation. Then bed preparation. So you have bed is does not mean for sleeping. So here the bed preparation is meant for the cultivation of these mushrooms, like how you are going to take a pot and put it in the put the soil where you're before sowing your seeds. In the same manner, here we are going to have some sort of bed preparation. So here the soil is going to be acting as a soil or substrate or compost, whatever the thing is going to be considered as a substrate, and that you are going to keep it in a proper manner to allow the growth of mushrooms. So that's all comes under the bed preparation. Then casing, how you are going to maintain the temperatures, humidity, all the things comes under the casing. Then allowing them to grow, and then your cropping, and after cropping obviously comes the harvesting, and then your yield, whether you got a good yield or you had a loss. All the things depends upon your proper following all these steps. Okay, so let's see each step one by one. And remember, here the production of mushroom spawn is going to be the similar for all the three types of mushroom that we are going to discuss. So listen in the first type, where I will go a little bit faster in telling about the spawn running in the rest of the two types. Okay, so let's go to the first step, production of mushroom spawn, and that too related to your Agaricus bisporus, but it is similar to other types of mushroom spawn also. To start growing mushrooms, you need to buy the spores. The spores shown below have been grown on a corn cob. So they have taken a corn cob on which uh, they are spread a little bit of the fungi, and it is going to have the spores on the right have been broken into 8 pieces. So these are the look. I'll tell you how to have the development of this spawn. Okay, now, so here is a spawn, what you are supposed to do. So you have to take a good quality of wheat grains. So obviously you know what are wheat grains, and then you have to clean them and dry wheat grains, and then you're supposed to soak it in water for about 8 to 10 hours. After soaking, you have to cook the wheat grains until they get very soft, okay, such that you can have the splitting of it very easily, and remove the excess water. Now spread these boiled wheat grains on a plastic sheet and add 4% calcium carbonate. So this will not allow the bacterial contamination. Okay, for that we are going to add this one. Then fill all the wheats containing this calcium carbonate mix in the bottles and plug with non-absorbent cotton. Now keep it for autoclaving purpose. That means if any spores or anything is are present also, they are also getting killed. Okay, after that, inoculation with pure culture. So we'll get some pure culture from somewhere, and that have to be inoculated in all the bottles that we have bought. and now you have to incubate at room temperature for 1 week. After 1 week, you are able to see the commercial spawn. So this is how they look like. So after 1 week, you are going to have a white meshy growth of this fungi. So that is going to be called as a spawn. Then coming to this, they are going to pack it. So this I'll tell you how they are going to pack all the things. So after taking the substrate, so here this is the substrate in which they have sprinkled the bottle spawn and they are packing. So after 12 to 14 days, you are able to show the pinning of these things and you will get the nice mushrooms from it. And this is the overall process of this button mushrooms, how they have grown. It's a pick. Okay, and now what you are going to do here is uh, the substratum for button mushroom cultivation. We are especially discussing about the button mushroom cultivation, so we are supposed to learn about this first one because this is being different from other agarical pleurotus, that is oyster mushroom and paddy straw mushroom, mainly in the stage of substratum. Okay, so here in this button mushroom we are using a compost. So what is this compost, what it is made up of? Here we are going to take, in India we can take either the wheat or the paddy straw, obviously waste. So they are made into a fine powder. Okay, so firstly the wheat straw is wetted for about 48 hours, and then wheat bran, the husk of it, is mixing with ammonium sulfate, superphosphate, urea, muriate of phosphate, or mixed in sawdust, and this is called as a fertilizer heap. And you are supposed to add the water for a day to this heap, and next day the mixture is mix it to the watered straw. That means in the previous day separately wheat straw and the wheat bran along, which we call them as a fertilizer heap, both are going to be combined now. Now this is going to be considered as a compost. So what are the two things that are present? One is the wheat straw and another one is the fertilizer heap were mixed and made into a compost. Now this compost should be turning. That means we have to mix it upside down, downside up. So those are being called as turning of compost. And this turning of compost have to be done on 6th day, 10th day, 13th day, and on the 13th day we have to add the gypsum. Again on 16th day and 19th day add 10 more kgs of gypsum, and finally the last turning is going to be done on the 22nd day. And now this we are ready with our compost and with our spawn. So two steps are over. What are those two steps? One is the spawn running and second one is the compost preparation. Now comes with the second step, third step, where you are going to have the bed preparation and then cropping. So for this what you are supposed to do is, compost is filled in the wooden trays. So we are going to take the trays here, wooden trays, and those wooden trays are going to be arranged one above the other, and those are pasteurized in the pasteurization room at a temperature of about 60 degrees centigrade and maintained for a period of about 2 days. You have to maintain it for 2 days, after which the trays are transferred to the production room. So we are going to have shifted these trays into the production room after pasteurization, and there the temperature will be only about 24 degrees because we have to allow the growth of mushrooms. Small pieces of commercially produced spawns are inoculated onto the compost bed. After spawning, the bed is covered with a neat, clean and water-wet newspaper. Remember, we are supposed to cover the trays with a clean, neat and water-wet newspaper. It is important that newspaper should always be wet. A good white cottony growth is observed on the compost surface below the newspaper after 10 to 15 days. I told you, after the 10 to 15 days we are going to have a small pinning or fungus like growth on it. Okay, the newspaper is removed after 20 days and now the trays are subjected to casing. What do you mean by casing? Casing denotes the covering of the bed with the soil. So here in the starting process I told you. So this is all the called as composting. So we had prepared the bed and it started off pinning. The mycelia have grown all the things and the spawn. This is the spawn. That is the inoculum of mushroom is being called as a spawn. And after once you see the fungi, you're supposed to have the layer of soil, which we are calling it as casing. Casing coil can be prepared with three parts ordinary soil and one part of sand, and it have to maintain at a natural pH. And here also we have to maintain the sterile condition. That means soil also have to be sterilized, and then it should be used in this casing process. Okay. Now, you have to maintain all the conditions of humidity, temperature, and everything. Then obviously you will have the rise of uh, pinning of heads of this, and then obviously finally you will have your button mushrooms. And these button mushrooms, excuse me, after full flushes of the thing after harvesting, the first flush will be taken up and the second flush will be appear in about 10 days, and third and fourth flushes will be appear after 8 to 10 days of the second flush. So that's how you are going to have your harvesting. And then mushroom should be harvested before watering only, and the lower parts have to be cleaned properly because the soil may be attached and where the quality may be damaged. So you're supposed to clean with a dry cloth, and then they are packed and they are going to be sent to the market either in the fresh manner or in the dried manner. So that's all about the button mushroom cultivation. So we have to take the compost, and then we have to have this processing of composting, and then you have to prepare the bed preparation. After the bed preparation you are supposed to add the spawn, and those are have to be run. That is, a cropping should be done. After cropping you are supposed to have the casing, and casing is going to be allowed. After casing you have to run the things, and once you get the first flush you'll have to take out the things, and then you have to go for the second flush, and after that you have to clean them, and you are supposed to do the processing, and then they are going to be marketing. So this is all about the overall cultivation of agaricus bisporus, okay, commonly called as button mushrooms. So let's move to the, these are all the things: canning, drying, freezing. These are the processing technologies where we can use to increase the shelf life of these mushrooms. Okay, so that's all about agaricus bisporus. So now we are moving into the second type, that is Pleurotus austerotis. What is the second type? It is Pleurotus austerotis. So this is how uh, this is a Pleurotus austerotis. Maybe I can tell you the Volveraceae. Then we'll move to the Pleurotus. So we will discuss this Volvarella volvaceae first, and then we'll move to the middle one, that is Pleurotus austerotis. So let's see the Volvarella volvaceae. The Chinese or paddy-straw mushroom, commonly called as paddy-straw mushroom, which are being widely used in the markets of especially in the tropical and subtropical regions of Asia, example China, India. Because why India and China are going to be the most populated density of countries, so they should have the plenty of food. So to replenish all the nutrients, they have to try the new things, where the mushroom is one of it. Okay, so now coming to this Pleurotus, Volvarella volvaceae, its cultivation. So as usual, all the practices will be similar in all the three: production of mushroom spawn, substrate, bed preparation, and then casing, cropping, harvesting, and yield. So let's see the first one, paddy spawn. I already told you what is a spawn. isn't it? So that this is the spawn production. So this is common for all the three types, so I'm not going again here. So here is the so here comes to the substrate that is differing from your agaricus bisporus, that is button mushroom. So here substrate, obviously we are discussing about a paddy straw, so the main constituent or substrate that we are using is a paddy straw. And some other country, some other places where they are having a wheat straw, maize straw, banana leaves, even banana leaves can also be used for this cultivation of paddy straw mushrooms. Okay, then coming to the bed preparation, the third, this one, bed preparation. Firstly, hand harvested 3 to 4 long inches, well dried, disease free paddy straw to be taken, and they are supposed to be bundled. Okay, that means we have to prepare the bundles with that straw. So like that we are supposed to prepare so many bundles depending upon our cultivation. So nearly one bed requires 35 bundles, and these bundles ought to be soaked in water for 8 to 16 hours and then taken out and washed with fresh water, and then you are supposed to drain off the excess water. Then that comes your bed preparation. Now a bed is prepared by putting 4 layers of this paddy straw bundles one over the other. Now the next one obviously is your adding the spawn. The spawn bottle is opened and racked with a woolen or glass straw and sprinkled by hand all over the margin of the bed, about 10 centimeters away from the edge, and continue up to 23 centimeters inside. The central portion of the lower layers should be left over. Okay, the sprinkled spawn is covered with a light dusting. Dusting means just not to sticky with the other thing, so you are supposed to have some dusting. Mainly here we will use the gram powder, that is your basin powder. Okay, this is the first layer. Then you are supposed to have the second, third and fourth layers are also prepared in the same way as in case of first. But the difference in the fourth layer is that sprinkling of spawn is done on the entire surface. In the bottom layer, that is first, second and the third layers, we did not sprinkle the spawn in the center. But coming to the fourth, that is the top layer, we are going to sprinkle the spawn entire surface, isn't it? The layer should also be dusted by the gram powder and must be covered with a thin layer of straw up to 8 centimeters thickness. This is all done with the remaining 3 bundles of the straws. Now we have done the bed preparation. Now we have done with the casing. Now we have to allow it for the cropping. Now water is sprayed for 2 to 3 times in a hot day and 1 to 2 times in a rainy season. If necessary, you can add some sort of insecticides, pesticides to decrease or to kill them. Next, coming to the cropping. Cropping starts 10 to 12 days after spawning and remains up to 15 to 20 days. Now coming to the harvesting. Mushrooms harvested at button stage or just after rupturing of the cup by twisting so that broken pieces are not left in the beds. Otherwise, bacterial rotting generally starts and spreads to the other healthy mushrooms. So we should not do that one. So after harvesting, that means eat, the mushroom should be used within 8 hours as I told you, or kept in refrigerator for 24 hours. Otherwise they'll get spoiled, and otherwise you can convert them into the dried form and you can market it and seal. After drying they are packed and sealed, otherwise they absorb the moisture which spoils them. So the packing of mushrooms is preferred only in the button state. They should have only in this button state. Whether if they are going to be in this stage, it is not going to be beneficial. Okay. So this is all about the common cultivation practices that follow in the second type of mushroom cultivation. That is, what is the second type? Volvaceae, which we commonly call as paddy straw mushroom. So just the difference here, if you see here, this is a paddy straw bundles. So they have wetted and they have prepared the bed bundles of having the 4 layers, and now they have added the spawning, and after mixing layer to layer they added the gram powder, and they pressed it, and the whole thing was covered in the plastic sheet. And after once the mycelium has observed, they are being shifted to the cropping room, and they are being having the fresh air, light, and the temperature is maintained. Once the fruiting is occurring, after fruitification they are harvested, and then it was going to be of marketed. So this is all about the paddy straw mushroom cultivation. So then again processing. So for the future use or the preservation, we can follow all these techniques. And in the food industry, they were used of having these techniques for the processing. So that's all about the second type of cultivation practices of paddy straw mushroom. So just here is a common pathway difference between the Agaricus bisporus and the paddy straw mushroom, that is button mushroom and the paddy straw mushroom. So here only the region where we are going to have is the compost. In the first Agaricus button mushroom, we use the compost, that means powder type of the thing, and here directly we use the straw beds are being used, and in both the cases spawning and rest of the things are going to be the same. Okay, I think so you understood. And we will move to the last type of mushroom, that is, what is it? Oyster mushroom, where commonly called as oyster mushroom, and it is a pleurotus species. Okay, this is a Pleurotus ostrotis. As I gave you a hint that here the ostrotis indicates the oyster mushroom, where it in India it is called as dinguri because it is having a spatula-like structure or an oyster shaped structure. So that's why they are being in different colors. Either it may be a yellow, brown, or creamy color, or white in color, or gray color, whatever it may be. Okay, and they majorly grow on the hard woods. So let's go with the cultivation practices of this also. So this is how they look like. Okay, and here the production of mushroom spawn. So obviously the same thing. Again, we'll take the wheat, we'll wash it, we'll soak it, and then we'll cook it, and we'll pour that cooked wheat grains in the bottles, and then we'll add the pure culture. We'll put the non-absorbent cotton plugs, and we will allow it further growth. And now we can run the we are having the ready of the spawn with the of uh, incubation. Okay, then coming to the substrate. Here the substrate that we are going to use is going to be of, what is the substrate that we are going to use here? Various substrates. So like wheat, paddy, straw, maize stalks, maize cobs, or cotton waste, etc. You can take off. Even wooden logs, sawdust, and vegetable plant residues can also be used for the cultivation of these. What are these called as? Yes, these are called as oyster mushrooms. Okay, then how you have to prepare the bed. So for the bed preparation, the paddy straw, you have to take the paddy straw, and it have to be chopped. It have to be chopped into 3 to 5 centimeter pieces and soaked in the water. So you are supposed to have the soaked in the water, that is in the fresh water, for about 8 to 24 hours. After that, these have to be drained off, that water have to be drained off, and that have to be kept in a gunny bag, and those gunny bags, either directly or by keeping in the gunny bags, you have to pasteurize it. That means these chopped paddy straw pieces were kept in a gunny bag, and that was kept dipped in a hot water of about 80 to 85 degrees centigrade for about 30 to 60 minutes. That's why we are calling it as pasteurization. Now, when pasteurizer substrate, now you have to stop the pasteurization, and let it cool. When pasteurization substrate has cooled down to room temperature, now it comes to the room temperature. Okay, it is ready for filling and spawning. Spawning means adding of the inoculum to the substrate is being called as spawning, or adding the spawn to the substrate is called as spawning. Now once it is having, at this stage the moisture content should be about 70%. Remember, the moisture content should be of about 70%. Now this mixture can be kept in the polythene bags like this, that is called as bagging. Okay, polypropylene bags or polythene bags, anything can be used. And then you are supposed to have the next step of running. So spot bags should be stacked in racks in neat and clean place in a closed position. So this is how they are going to do. So loose closely, closing the mouth of polythene bags filled with spawn compost. Shifting the compost filled bags in cropping rooms with a temperature of 23 plus or minus centigrade, and room temperature, that is humidity, should be of about not temperature, humidity should be of about 95%, and the carbon dioxide concentration should be of 1 to 1.5%. And keeping the bags under above condition for about 12 to 14 days, then completion of the spawn running is going to occur. Once bags are fully covered with the mycelium, they are transferred to the crop room and polypropylene covers are removed. That means these covers are being removed or removed. Now, it grows in a temperature, I told you about 23 plus, and then it maintain of temperature, room humidity is going to be of 80 to 85%, and then you're supposed to spray the water for every twice a day, but watering on blocks should be avoided for first 2-3 days. That means we have to spray the water only on the walls but not on the directly on the beds. After 2 to 3 days, light mist spray of water is given on blocks. This is the time when small pinheads appear. So like this you can see, so these are the pinheads. Once pinheads are of 2 to 3 centimeter size, a little heavier water is done on blocks and further water is stopped to allow them to grow. Once mushrooms are going to be of 6 to 8 centimeters in size, they are plucked. Mushrooms should not be allowed to head spores as it results in poor quality of mushrooms. Obviously, if they are going to be of spore production, we won't use it in the consumption. After harvesting the first flush, as I told you, uh, you are supposed to remove the outer layer which are having the scrapes of the first flush. This helps to clean remnants of the first crop and to initiate second flush. The second flush will appear in about 10 days of first flush. Similarly, you can have the third and fourth flush will appear in 8 to 10 days interval. But majority of the harvesting people, that means mushroom cultivating people, will take only the first and second flushes. Hence, many growers will uh, opt only the first two flushes. Okay, now you are going to have the harvested mushrooms with you. So mushrooms should be harvested. When to harvest? Only they should be harvested before watering, and their lower portion is cleaned with dry cloth. They are packed in perforated polythene or polypropylene bags. It should be sent to the market while fresh, or they can be sun dried by keeping fresh mushroom in sun for 2 days. Or nowadays we are having the mechanical dryers uh, where you can have the new technology can be used for the preservation of the longer preservation. So here are the processing technologies where canning, drying, freezing, freeze drying, chemical preservation, pickling, steeping preservation. So by all these techniques, the mushrooms can be preserved for a long time. So that's all about the different types of mushrooms that are being cultivated in India. Okay, I hope so you all understood. Thank you.